Hello students, welcome to EPG Pathshala. I am Dr. M. Gupta, Emeritus Professor at Department of Biochemical Engineering and Biotechnology at Indian Institute of Technology, Delhi. From today, we will start talking about the lipids, and in today's module, we are going to introduce lipids an introduction, and the paper of course is structure and function of biomolecules 2. For quite some time, lipids were considered not as exciting a class of biological molecules as proteins and nucleic acids. Nucleic acids are genetic material, so they attracted the attention of mainstream biochemists even prior to recombinant DNA era. Enzymes were proteins and catalysts of all the reactions in living organisms is carried out by enzymes. So they were the focus of microbiologists, biochemists, and many others. Lipids in the beginning were of interest mostly just to the nutritionists. Now, of course, the general population is also interested in fats and oils and omega fatty acids in view of the greater exposure to some of these issues, the way they are mentioned in the media, and also in view of or as a result of massive advertisement by the oil industry in the media. However, the role of the lipids goes beyond nutritional aspects. Today, let us look at a brief overview, and we will cover their various biological roles in this set of modules, which are exclusively devoted to lipids, the various classes of lipids. So, the objective of this module is straight away to understand the general importance of lipids in biology and biotechnology. With more than $1 billion US dollar market, products of the oleochemical industries touch our daily lives in many ways. Somehow the biotechnological roles, especially of lipids, is not generally emphasized by general textbooks of biochemistry, and so we will also mention some of these roles. So the concept is essentially about understanding that what are lipids made up of. Carbohydrates are sugars, they are polymers and other derivatives. Proteins are made up of nucleic acids. Nucleic acids are made up of nucleotides. So what are lipids made up of? Structurally speaking, lipids is a very heterogeneous class of molecules. All the classes of biological molecules discussed so far had a structural basis. Lipids differ widely in their structures. As we will see, structure of a fatty acid has little resemblance to a cholesterol molecule. Both, nevertheless, are classified as lipids. To start with, both show higher solubility in organic solvents than in water. So, it is actually a physical property of solubility which was responsible for both and many other substances being classified together as lipids. Interesting enough, the later work showed that fortuitously this turned out to be a judicious choice. Cholesterol and lipoproteins, for example, turned out to have a close relationship in metabolism. So look at the clinical reports on the blood of the people, and if this report includes what is known as the lipid profile, so you would find there the triglyceride content, cholesterol, fine, but you will also find low density lipoprotein and high density lipoprotein all mentioned together. So it turned out that biologically it was not a bad idea to classify these different kinds of molecules all as lipids. Historically, the class of lipids emerged as any compound which was part of a fraction which was extracted from a biological source. For example, an animal source or a plant source. The extraction was carried out by so-called fat solvent, which are actually non-polar solvents like benzene, ether, chloroform or petroleum ether, etc. So the basis for compounds being classified together is operational in this case rather than the structural one. In order to understand why it happened this way, it should be remembered that the early biochemists had to heavily rely upon the techniques used by organic chemists. For understanding what all are present as components of the biological materials, the biochemists heavily relied upon extraction as the tool of isolation in those early days. This problem is not limited to the lipids. For example, even in the case of the proteins, the first classification of proteins thus was based upon extractability or solubility. Thus, as you see in the slide, the classification of the proteins in those days consisted of albumins, which were soluble in water and salt solution, globulins, which were barely soluble in water but freely soluble in salt solutions. Prolamines were soluble in 70 to 80% ethanol. Then there were glutalines, which were soluble in acid or alkali. Finally, there were scleroproteins, which were insoluble in aqueous solvents. In the case of the lipids, it gradually emerged that what gets extracted may depend upon the nature of the organic solvent. For example, a polar organic solvent may extract selectively a different constituent of the biological material. If a more polar or less polar organic solvent is used, what may get extracted as a lipid material may have a different composition. This is the situation which led to the understanding that the compounds extracted with organic solvents are actually a heterogeneous class. Let us look at the various subclasses of lipids. The simplest subclass of the lipid consists of fatty acids and also the lipids which are derivatives of the glycerol. Glycerol, as you probably already know, is a trihydric alcohol. In daily life, it is used as a moisturizer. Industry uses it as a solvent. it is commonly called glycerine. Glycerol is freely soluble in water. Acidifying its alcoholic group with long chain fatty acids turns glycerol into fats or oils. During soap making, a process which has been generally called saponification for a long time, glycerol actually is produced as a byproduct. Currently, most of the industries which were major suppliers of glycerol in the world are shut down. These days, most of the glycerol which is available in the market is a byproduct during biodiesel production. In a later module, we will see what biodiesel is and how glycerol arises as its byproduct. The next subclass of the lipids consists of neutral fats and are actually what we generally call fats or oils. Structurally, both fats and oils are actually identical. If the triglyceride is present as a free-flowing liquid, we call it an oil. However, if this is present as a solid, we normally call this a fat. While something like lard will be solid at wide range of temperatures, which may be prevalent as room temperature in different seasons or in a different country, there are other triglycerides which can be present both as a solid or a free-flowing liquid depending upon the ambient temperature. In a country like India, where the temperature vary widely in different seasons, this may get slightly confusing. For example, in Delhi winter, coconut oil is generally solid. In other seasons, it may be semi-solid. As we will learn in a later module, there is a process in oleochemical industry which is called winterization. Winterization consists of cooling an oil and fat at extremely low temperature and removing the solid fraction of the oil. In general, we should not get confused by this terminology. Normally, a standard temperature of 25 or 30 degree centigrade can be selected and an oil can be labeled as an oil if it is a free-flowing liquid. While triesters of glycerol, fats and oils are well-known compounds, glycerol also forms another important subclass of lipids. These are the ether derivatives. Here are examples of one of its terminal carbon atom as a part of ether linkage. The examples which are given here are salicyl alcohol, schimmyl alcohol and betyl alcohol. Please note that the remaining two OH groups are free in these compounds. These free OH groups, which are a part of the glycerol chain, make these compounds fairly polar. For this very reason, these compounds are actually called alcohols. The long chain R of the other component of the ethyl glycerol, OR, ensures poor solubility of these compounds in water. On the other hand, it also imparts solubility in organic solvents to these compounds. For this reason, the glycerol ethers like salicyl alcohol, schimmyl alcohol, betyl alcohol are also classified as lipids. The next important subclass of lipids is phosphatides. These are also classified under glycerophospholipids. As can be seen from the slide, in these compounds, the nitrogenous compounds choline, ethanolamine and L-serine etc., which contain the alcohol group, forms ester bonds with the free acidic group of the phosphoric acid of the L-phosphatidic acid, and that is why these are generally called glycerophospholipids. You may have noted that we call this isomer L isomer, and that is because of its stereochemical relationship with L-glyceryl phosphate. There is another way to look at the phosphatide structure. This structure can be looked at as monophosphate ester of 1,2-diglycerides. When the phosphate group forms another ester bonds with these nitrogenous bases containing alcohol, many important phospholipids are formed. As we will see in a later module, glycerophospholipids are the phospholipids which are generally present in biological membranes. Hence, glycerophospholipids are of tremendous biological importance. Phospholipids like phosphoglycerides are derivatives of glycerol phosphate and do not contain nitrogen. All these are, as we just learned, are also called phosphatidic acids. The common phosphoglycerides present in animals and plants are diphosphatidylglycerols glycerols and monophosphatidylglycerols glycerols respectively. So it is necessary to realize that phosphatidic acid is not a single compound, but a class of phosphoglycerides. Different members have different fatty acids, acidifying the OH on the glycerol chain. Phosphoinositides are derivatives of alpha phosphatidic acid without nitrogen but contains inositol. Inositol is a sugar alcohol. Myoinositol is the best known member of this group as it is widely distributed among microorganisms, plants and animals, often as a phosphorylated form. In animals, myoinositol also occurs in free form in tissues like muscle, heart, lung and liver. The structure of the myoinositol is shown in the slide. Just to clarify again, currently phosphatides, phosphoglycerides and phosphoinositides are all considered members of a bigger class which we have already called glycerophospholipids. Glycerophospholipids are also excellent surfactants. Dipalmitoyl phosphatidylcholine, generally abbreviated as a DPCC, as we will see, is an important lung surfactant required for good pulmonary performances. Next, let us look at the sphingolipids. Sphingolipids are also part of membranes. In most sphingolipids, sphingosine which is a C18 amino alcohol, has trans configuration. The trans configuration is shown in the structure of the sphingosine, which is around its single double bond. In dihydrosphingosine, of course, this double bond is abolished because after hydrogenation of the sphingosine, the dihydrosphingosine is created. Sphingosine, please note, has two OH groups and one amino group as main functional groups. It may be interesting to look at the biosynthesis of sphingosine. The biosynthetic route is shown in the slide. Apart from these most common C18 sphingosines, less common C16, C17, C19 and C20 sphingosines also occur as a part of some naturally occurring sphingolipids. As we see, basically sphingosine is biosynthesized starting from palmitoyl-CoA and serine, and the ultimate product requires couple of other reduction and other kind of a chemical reactions. The next important class of sphingolipids are ceramides. Ceramides thus are sphingolipids in which the free amino group of the sphingosine is acylated by a C16, C18, C20 or C22 fatty acids. Thus, ceramides have no free amino group left. Thus, ceramides are essentially N-acyl sphingosine. Ceramides differ from each other in this constituent fatty acids because this is the fatty acid which has acylated the free amino group of the sphingosine. Ceramides are the basic structural units of sphingolipids. Thus, ceramides are part of both sphingomyelin and gangliosides, which are two important classes of the sphingolipids. Sphingomyelins, for example, are important constituents of brain lipids and are also found in the blood. Plasma membrane of the animal cells contain sphingomyelins. Structurally speaking, as is seen in the slide, sphingomyelins are phosphodiesters of a ceramide and choline. The other important class of the sphingolipid is cerebrosides. Cerebrosides are ceramides which can be called glycosphingolipids. That is because they also contain carbohydrate as a constituent. The common cerebrosides are galactocerebroside and glucocerebroside. Here, the galactose or the glucose is linked to the terminal OH group of the sphingosine unit of the ceramide. Please remember that the sphingosine also had a free OH group in its structure. Also, it is to be noted that cerebrosides do not contain phosphate group and hence are neither phospholipids nor ionic in nature. So, sphingomyelins are phospholipids, whereas ceramides and cerebrosides are not. Let's continue to look at some important cerebrosides. Kerosene contains saturated C24 lignoceric acid in the ceramide part. Then we have a phrenosine This contains cerebronic acid, which is actually the 2-hydroxy derivative of lignoceric acid, and cerebronic acid is in place of just the lignoceric acid. Then we have a sulfatides. These cerebrosides are actually a group of sulfur containing cerebrosides. Here often sulfur is present as sulfoesters in the sugar part. We also have ceramide oligosaccharides. These contain more than one sugar residue, and depending upon how many sugar residues are present, they are called cerebroside disaccharides, and so on and so forth. Some example of cerebroside oligosaccharides are ceramide lactoside, which obviously is a cerebroside disaccharide. Another important example is cytolipine K, which actually has a tetrasaccharide moiety. The formula of the lactosyl ceramide is shown here, so you can see the linkages between the sugars and the other part. Let us look at the synthesis of gangliosides, which are another important class of complex lipids. We had mentioned earlier that the gangliosides are also formed from ceramides. Again, please note that just like cerebrosides, gangliosides do not contain phosphorus. So these are not phospholipids but are generally classified under glycolipids. Myelin sheath are membranes wrapped around each nerve axon. About 10 to 15 layers thick, it has a high lipid content and hence is an electrical insulator. Myelin sheath is rich in sphingomyelins. The name sphingomyelin, in fact, owes its origin to this fact. Most complex sphingolipids are also ceramide oligosaccharides but deserve to constitute a class of its own. Gangliosides have a specific structural unit inasmuch as the oligosaccharide chain in gangliosides contain an acidic sugar called sialic acid. Sialic acid also has another name which is called N-acetyl neuraminate. Its structure is shown in the slide. The so-called grey matter in the brain is rich in gangliosides. There is a disease called Tay-Sachs disease, which is generally fatal for infants. This is an inherited disorder associated with the deficient hydrolase, which breaks down a ganglioside. Now, let us look at another class of lipids which are called waxes. Waxes are esters of long chain fatty acids with long chain alcohols. So while fats and oils are esters of glycerol, please note waxes contain long chain alcohols such as cetyl alcohol. The structure of the cetyl alcohol is shown in the slide. Other important classes of lipids are terpenes and steroids. Many essential oils are terpenoids. Vitamin A, the visual pigment, and many plant pigments are terpenoids. Thus again terpenoids constitute a very heterogeneous class. Similarly, steroids is another heterogeneous class and consists of variety of molecules with different physiological properties. Steroids are compounds having cyclopentane perhydrophenanthine fused ring system. This fused ring system is of the steroid is shown in the slide. There are some important steroids, for example, cholesterol, bile acids and sex hormones are important examples. So students, let us now summarize what we have learned in this module. We learned that all compounds soluble in organic solvents and mostly insoluble in aqueous buffers are called lipids. So structurally, this is a very diverse class. This, as we shall see, results in lipids having diverse roles and functions in biological systems. Broadly, we have also gained preliminary knowledge about these various classes of lipids. Fats and oils top the list. Phospholipids, Spingolipids, waxes, terpenoids and steroids. And we also got a glimpse about the fact that the lipids also combine with other classes of biological compounds to form lipoproteins and lipopolysaccharides. Thank you.